All right, so today we are deep diving into a topic that is, well, it's pretty essential to, well, to life as we know it.
Absolutely. Antibacterial drugs. We're
Mm.
taking a look at the sources you've provided, and they actually lay out a really good kind of like structured overview of the main categories of these medications.
Yeah, I mean, it's a fascinating, right? Like we've got these, these different kinds of drugs that target bacteria, but it's like, how do they actually work? And then, we've got this huge issue of resistance, right? Like how do bacteria become resistant to these drugs? And I think what we want to do today is really kind of extract like the crucial information about, the mechanisms of action, how they work, and this whole idea of resistance, and try to do it in a way that's clear and concise, right? Like
Mhm.
we don't want to, overload you with too much information.
Right. Yeah, I think the sources you provided do a really good job of breaking down, like you said, the major classes of antibacterial drugs.
Yeah. So why don't we just jump right in, right?
Yep.
I mean, we've got let's see, we've got beta-lactam antibiotics, aminoglycosides, tetracyclines, macrolides, glycopeptides, sulfonamides, quinolones or fluoroquinolones, lincosamides, and oxazolidinones. It's a mouthful.
Yeah, that's a lot.
So let's start with well, how about beta lactams,
Mm.
right? I mean, everybody's heard of penicillin, right? Like how does that actually work? How does it stop an infection?
So beta lactams, they work by interfering with the bacteria's ability to build their cell walls. So you can think of it as preventing them from building this protective outer layer.
Right. So it's like you're building a house and you can't put up the walls, right?
Exactly. Yeah. So and that makes the bacteria vulnerable. They can't maintain their structure, and eventually they burst.
So beta lactams, they're pretty broadly used, right? Like I know they're used for skin infections, respiratory infections,
Yeah, yeah, very common for those kinds of infections, and a lot of other infections too.
and then we have aminoglycosides, right?
Yeah.
What do they do?
Yeah, so aminoglycosides, they disrupt the bacteria's protein-making machinery. So, they're essentially, you can think of it as throwing a wrench in the gears of the factories that are producing these essential components that the bacteria need to survive.
So it's like sabotaging the factory that makes all the parts for the bacteria to live, right?
Exactly. And these are often used for, more serious infections, things like sepsis.
Okay, and then we have tetracyclines.
Hmm.
I know those are used for a lot of different things, right? Like skin infections, respiratory infections.
Yeah, so tetracyclines, they also inhibit protein synthesis, but they bind to a slightly different part of the bacteria's protein-making machinery. So again, disrupting that crucial process.
Oh, I see. So it's like they're attacking the same factory, but they're going after a different part of it, right?
Yeah, yeah, that's a good way to think about it.
Now I know resistance can be a problem with tetracyclines, right?
Yeah.
Like the bacteria can figure out ways to get rid of them.
Yeah, that's right. One of the ways that bacteria can become resistant to tetracyclines is by developing mechanisms to pump the drug out of the cell. So, they're basically just spitting it back out.
So it's like they're developing like a little pump to get rid of the antibiotic.
Exactly.
That's amazing. And then we have macrolides. I think everybody's heard of erythromycin, right?
Yeah. So macrolides, they also target protein synthesis, and they bind to yet another spot on the bacterial ribosome. So again, highlighting just how crucial this protein synthesis process is for bacteria.
So it's like they're going after the same factory, but they're finding like a different door to get in,
Yeah,
right?
yeah, exactly. And these are often used for, things like a respiratory infection, some sexually transmitted infections, and resistance can occur by the bacteria modifying the binding site. So, they're basically changing the lock on the door.
Wow. And then we've got glycopeptides, right? Like vancomycin.
Yeah.
That's a big gun, right? Like that's used for really serious infections.
Yeah, yeah. Vancomycin is definitely a powerful antibiotic. And glycopeptides, they also prevent cell wall construction, but they block a different building block compared to beta lactams. So, if you think about it as disrupting the assembly line, they're just targeting a different stage in that process.
Oh, okay. So it's like they're both trying to stop the house from being built, but they're going after different parts of the construction process.
Exactly. And vancomycin is often a crucial drug for serious resistant infections, when other antibiotics have failed.
And then we have sulfonamides.
Yeah, so sulfonamides, they interfere with the bacteria's ability to produce folic acid. And folic acid is something that bacteria need to grow and multiply. So by blocking this pathway, they're essentially preventing the bacteria from reproducing effectively.
So it's like cutting off their food supply, right?
Exactly. And these are often used for urinary tract infections. And resistance can develop, through mutations that allow the bacteria to bypass the need for the blocked pathway.
And then we have quinolones or fluoroquinolones,
Hmm.
which I think a lot of people are familiar with.
Yeah. So these target the enzymes that bacteria need to copy their DNA. So by inhibiting these enzymes, they're basically preventing the bacteria from replicating their genetic material.
So it's like stopping them from making copies of themselves, right?
Exactly. And these are used for, a variety of infections, including respiratory and urinary tract infections. And resistance can arise through mutations in the targeted enzymes.
And then we have lincosamides.
Right. So lincosamides like clindamycin, they again inhibit protein synthesis, and they attach to a similar site as macrolides on the ribosome. And these are used for things like skin and soft tissue infections.
It's just amazing to me how many different ways we can disrupt this protein synthesis process, right?
It's really remarkable.
Yeah. And then finally we have oxazolidinones.
Mm.
I think those are kind of a newer class of antibiotics, right?
Yeah, that's right. So oxazolidinones also block protein synthesis. But they bind to a unique spot on the ribosome, different from all the others. And this can be really important when bacteria have become resistant to other protein synthesis inhibitors.
Oh, so it's like they're finding like a whole new way to attack the factory, right?
Yeah, exactly. And they're used for some, serious infections, particularly those that are resistant to other drugs.
So I mean, we've talked about all these different classes of antibiotics and how they work, but I think one of the biggest challenges that we're facing now is antibiotic resistance, right? Like how do these bacteria actually become resistant to these drugs?
Yeah, and understanding these different mechanisms is key to understanding how resistance develops. So, for each target that a drug hits, bacteria can potentially evolve a way to counteract that effect.
Hmm.
Whether it's by producing enzymes that break down the drug, modifying the target site, finding ways to pump the drug out. The selective pressure from antibiotic use really drives this evolution.
So I mean, it's almost like an arms race, right? Like we're developing these new drugs, and then the bacteria are figuring out ways to get around them.
Exactly. Yeah, it's a constant battle.
So I mean, for you, the listener, I think what you should take away from this deep dive is that we've got these major categories of antibacterial drugs, and they work in very specific ways to disrupt bacterial life processes, right? And we've talked about some of the infections that they're used for. But I think it's really important to understand that there's this fundamental link between how these drugs work and how resistance emerges.
And I think, the sources that you provided really do a great job of providing this framework for understanding this complex and constantly changing field.
Yeah, and so kind of like a final thought here, given the diverse array of targets and mechanisms that we've discussed, what are some of the potential avenues for developing the next generation of antibacterial drugs? Like, how do we stay ahead of this evolving bacterial threat?
Yeah, that's the $1 million question,
Hmm.
right? And there are a lot of different approaches being explored, but it's definitely a complex challenge that's going to require a lot of innovation and collaboration.
Well, on that note, thanks for joining us for this deep dive, and hopefully you've learned something new today.
Yeah, thanks for having me. It's been a great discussion.
All right, take care.
You too.